everyone, this is Himanshu, and in this lecture, we will study more about the gene editing technique. Now, the gene editing is the process of disabling the faulty genes and correcting the harmful mutations by inserting a missing gene or the changing the activity of the specific genes in a living organism. So, this is what generally happens in the gene editing. So now, because of this, it has multiple applications. Applications like it helps in treating the genetic disorders, and gene editing is actually widely used to modify, the immune cells. So it helps in modifying the immune cells. And it also helps in preventing the inheritance of the genetic disorders. So prevent inheritance of genetic disorders. And more importantly, gene editing techniques are actually the faster, cheaper, and more precise than the conventional technique, like for example, like genetic modification of the crops. This is actually much better from those. And further, the gene editing techniques are extremely helpful in the gene therapy, including the building of new diagnostic tools for the drugs and the organ transplantation. So this is what generally happens in the gene editing. Now, recently there was also in the news about the in vivo gene editing. So it was something like this: in vivo gene editing. Now, recently the gene editing was carried out inside the human body. So when we say when it is inside the human body, now this is what we call as the in vivo. So if I highlight only this word, that let me write it like this. Now if I say this thing, this means that inside human body. Now, when the gene editing was carried inside the human body for the first time to treat the Hunter's syndrome, so this is what we call the in vivo gene editing. The zinc finger nuclear ZNF technique was actually used in this thing. So you can keep in mind that ZNF, that is a zinc finger nuclease technique was used here to insert the corrective gene in the DNA of the patient through the intravenous therapy. That's an IV. So this is what generally happened in the in vivo gene editing. So I think this is the pretty much good information that you can remember. You don't have to remember anything more than this. Next one is basically the CRISPR Cas9. CRISPR Cas9. Now this is basically the one of the mechanism from where we do the gene editing itself. So it is a gene editing technique. Gene editing technique. So basically, if you want to know the full form, it is something like this: like clustered, regularly interspaced, short palindromic repeats. You don't have to remember that. The CRISPR Cas9 is good. So the CRISPR, actually there are two things, CRISPR and the Cas9. So the CRISPR uses an enzyme called Cas9 to cut the strands of the DNA at precisely targeted locations and insert a new genetic material into the gap. So if I say that if this was a helix, so what generally happen here, that I will use a CRISPR technique, and through that I will remove this part, which is basically the faulty genetic part, you can say, on a targeted locations. I will remove this thing and replace it with this thing. Okay, and this whole procedure, now this what I'm adding, this is basically an enzyme, and this is called the Cas9 enzyme. This is a Cas9, and this is basically the CRISPR. So this whole procedure of removing a particular strand of the DNA, which is basically a faulty one, by a new one, so that it can function again properly, this whole procedure is called CRISPR Cas9. The CRISPR is a replica of the natural process when a virus actually infects the bacterium. So the bacterium has evolved the CRISPR Cas9 system, adaptive immune system. So this was actually main logic behind this. So this is mainly about the CRISPR Cas9. Let's jump to the next one. The next one that we're going to talk about is the Gene Drive Technology. Now, what generally happened that, in a breakthrough in the global fight for the malaria, the scientists have wiped out the entire populations of the malaria-carrying mosquitoes in the lab condition using the CRISPR-Cas9 gene driving technology. So, gene driving technology is what we consider as a genetic engineering technology, because name itself says this thing. So, it is basically genetic engineering technology that can permanently change the traits of the population or even an entire species, and that's what it makes it dangerous. So it can permanently change the traits of a population or even an entire species. So this is what we call as a gene driving technology. Now, the gene drives are genetic elements that passes from the parents to unusually high numbers to their offsprings, and thereby it spreads quickly. So this is what it is. So the gene drives occur naturally, but it can also be engineered. So if you see that how does it work, so the gene drive technology is basically designed to introduce a genetic tweak in a population of a species by altering the rules of the inheritance from the parent to the offspring. So if this is a parent, and okay, fine, right? So now we can say that these are the further offsprings of that, right? Then and so on, the cycle continues. So I'll do some tweaks here, and it will automatically have an inheritance over all of them which are under them. So this generally what happened. So first, using the CRISPR Cas9, so let's make it here like the first, we will use a CRISPR technology here, which is a gene editing tool, and we will, the example of the mosquito suits fine to me. So these are all, just imagine these are mosquitoes, right? So first what we're gonna do, we will do some gene editing, and a gene which is called the double sex in the female mosquito, that will be disrupted. So using CRISPR Cas9, we will disrupt the chromosome, or you can say disrupt a gene that is called the double sex. Now, because of this genetic tweak, it leads to a different inheritance, means it will also be followed in this one, and here the gene drive inheritance make the female mosquitoes inherit the 2 copies of the disrupted genes in each and every level. Now, when the female mosquitoes inherit 2 copies of the disrupted genes, they develop like males and are unable to beat or lay eggs. Understood? And because of that, they can no longer lay eggs, and this is how the malarial mosquitoes can be tamed. So there won't be a malarial mosquito. So in short, we will do a genetic mutations, and because of that, it will disrupt the cycle and we are changing the way of the nature how they are governed. But through the CRISPR Cas9 technology, we are wiping out the population of the malarial mosquitoes. So this is what the gene drive technology is. It doesn't stop at a one particular level. It leads to the further inheritance, and it has the capability of completing wiping out entire species. So that's why we call it gene drive technology. So this is mainly it about. So this is it about this lecture. In the next lecture, we will study more about the GM crops. Thank you guys.